I am Mary Luz Funtecha.
I am Lester Mike Brigado.
I am Ariane Lanoy.
I am Tatabai Kanda.
Lipids and membranes. Lipids are nonpolar compounds soluble in organic solvents. Most membrane lipids are amphipathic, having a nonpolar end and a polar end. Fatty acids consist of hydrocarbon chain with a carboxylic acid at one end. Double bonds in fatty acids usually have the cis configuration. Most naturally occurring fatty acids have an even number of carbon atoms. So there's the sum of fatty acids and their common names. First, myristic acid, palmitic acid, stearic acid, oleic acid, linoleic acid, linolenic acid, arachidonic acid, and icosapentaenoic acid. There is free rotation about double carbon bonds in the fatty acids hydrocarbon except where there is a double bond. Each cis double bond causes a kink in the chain. Rotation about other double carbon bonds would permit a more linear structure than shown, but there would be a kink. Glycerophospholipids. Glycerophospholipids are common constituents of cellular membranes. They have a glycerol backbone. Hydroxyls at C1 and C2 are esterified to fatty acids. An ester forms when a hydroxyl reacts with a carboxylic acid with loss of H2O. Phosphatidate. In phosphatidate, fatty acids are esterified to hydroxyls on C1 and C2. The C3 hydroxyl is esterified to P. In most glycerophospholipids, P is in turn esterified to hydroxy of polar head group. Simply gracia, serine, choline, etanolamine, glycerol, or inositol. The two fatty acids tend to be non-identical. They may differ in length and the presence or absence of double bonds. Phosphatidylinositol with inositol as polar head group is one of glycerophospholipid. In addition to being membrane lipid, phosphatidylinositol has roles in cell signaling. Phosphatidycholine with choline as polar head group is another glycerophospholipid. It is a common membrane lipid. Each glycerophospholipids includes a polar region, glycerol, carbonyl O of fatty acids, P, and the polar head group, non-polar hydrocarbon tails of fatty acids. Sphingolipids are derivatives of the lipid sphingosine, which has a long hydrocarbon tail and a polar domain that includes an amino group. Sphingosine may be reversibly phosphorylated to produce the signal molecule sphingosine 1-phosphate. Other derivatives of sphingosine are commonly found as constituents of biological membranes. The amino group of sphingosine can form an amide bond with the fatty acid carboxyl to yield a ceramide. In the more complex sphingolipids, a polar head group is esterified to the terminal hydroxyl of the sphingosine moiety of the ceramide.
Cerebroside and ganglioside, collectively called glycosphingolipids, are commonly found in the outer leaflet of the plasma membrane, bilayer with their sugar chain extending out from the cell surface. Sphingomyelin with a phosphocholine head group is similar in size and shape to the glycerophospholipid phosphatidylcholine. depending on the lipid, possible molecular arrangements, various micelle structures, e.g. A spherical micelle is a stable configuration for amphipathic lipids with a conical shape such as fatty acid. A bilayer, this is the most stable configuration for amphipathic lipids with a cylindrical shape such as phospholipids.
Cholesterol is an important constituent of cell membrane, has a rigid ring system and a short branch hydrocarbon tail. Cholesterol is largely hydrophobic, but it has one polar group, a hydroxyl, making it amphipathic.
Cholesterol is abundant in membranes such as plasma membranes that include many lipids with long-chain saturated fatty acids. In the absence of cholesterol, such membranes would crystallize at physiological temperatures. The inner mitochondrial membrane lacks cholesterol but includes many phospholipids whose fatty acids have one or more double bonds which lower the melting point to below physiological temperature.
Flip-flop of lipids from one half of a bilayer to the other is normally very slow. Flip-flop would require the polar head group of a lipid to traverse the hydrophobic core of the membrane. The two leaflets of a bilayer membrane tend to differ in their lipid compositions. Flipases catalyze flip-flop in the membrane where lipid synthesis occurs. Some membrane contains enzymes that actively transport particular lipid from one monolayer to the other. Membrane proteins may be classified as peripheral, integral, having a lipid anchor. Peripheral proteins are on the membrane surface. They are water-soluble, with mostly hydrophilic surfaces. Often peripheral proteins can be dislodged by conditions that disrupt ionic and hydrogen bond interactions. For example, extractions with solutions containing high concentrations of salt, change of pH, and chelators that divalent cations.
Good morning, ma'am and classmates. My name is Tata Bekanda and I'm going to tell you about Ag-plextrin homology domains bind to phosphorylated derivatives of phosphatidylinositol. Some pH domains bind PiP2, Pi-4, 5-P, and 2. Other plextrin homology domains recognize and bind phosphatidylinositol derivatives with Pi esterified at the 3-OH of inositol. Ag. Pi-3 P-I-3 4-P2 Pi-3 4 5-P and 3. Some proteins bind to membranes via a covalently attached lipid and anchored that inserts into the bilayer. A protein may link to the cytosolic surface of the plasma membrane via a covalently attached fatty acid E and G, palmitate or myristate or an isoprenoid group. Palmitate is usually attached via an ester linkage to the tail of a cysteine residue. A protein may be released from plasma membrane cytosol via depalmetylation hydrolysis of the membrane. An isoprenoid such as a farnesyl residue is attached to some proteins via a thioate linkage to a cysteine thiol. GPI are complex glycolipids that attach some proteins to have the outer surface of the plasma membrane. The linkage is similar to following O2. The oligosaccharide composition may vary. Protein C-term and hotanolamine, manus, and acetylglucosamine and inositol of BSN membrane. The protein is tethered some distance out from the membrane surface by the long oligosaccharide. Integral proteins have domains that extend into the hydrocarbon core of the membrane. Often they span the bilayer. Intramembrane domains have largely hydrogen surfaces that interact with membrane lipids. Ampihepatic detergents are required for solubilization of integral proteins from membranes. Hydrophobic domains of detergents substitute for lipids, coating hydrophobic surfaces of integral proteins. Polar domains of detergent interact with water. If detergents are removed, purified integral protein to aggregate and come out of solution. Their hydrophobic surfaces associate to minimize contact with water. Lipid wraps are discrete lipid domains present in the external leaflet of the plasma membrane. The wraps are increased in cholesterol, glycosylphospholipids, and glycosylphosphatidylinositol anchored proteins and are insoluble in low concentrations of nonionic detergents. Complex sphingolipids tend to separate out from glycerophospholipids and glycolyze with cholesterol in membrane. Microdomains are called lipid wraps. Complex sphingolipids may be formed by addition of head groups to ceramide or phytoceramide. Sphingomyelins have a phosphocoline or phosphatidylamine molecule with an ester linkage to the 1-hydroxy group of differences in molecular shape may contribute to a tendency for sphingolipids to separate out from glycerophospholipids in membrane microdomains. Sphingolipids usually lack double bonds in their fatty acid chains. Glycerophospholipids often include at least one fatty acid that is inked due to one or more double bonds. See diagram in article. Lipid wrap domains tend to be thicker than adjacent membrane areas, in part because the saturated hydrocarbon chains of sphingolipids are more. Proteins involved in cell signaling often associate with lipid wrap domains. Otherwise, soluble signal proteins often assemble in complexes at the cytosolic surface of the plasma membrane, in part via insertion of attached fatty acyl or isoprenoid lipid anchors into wrap domains. Integral proteins may concentrate in rough domains via interactions with rough lipids or with other rough proteins. Some rough domains contain derivatives of phosphatidylinositol that bind signal proteins, plextrin, homology domains. Cavuli are invaginated lipid wrap domains of the plasma membrane that have roles in cell signaling and membrane internalization. Cavulin is a protein associated with the cytosolic leaf plate of the plasma membrane in cavuli. Cavuli interacts with cholesterol.
Integral protein structures are also called intrinsic proteins, have one or more segments that are embedded in the phospholipid bilayer. Atomic resolution structures have been determined for a small but growing number of integral membrane proteins. X-ray crystallography has been the most used method to determine atomic structures of membrane proteins, but it is hard to crystallize for X-ray analysis. Because of their hydrophobic transmembrane domains, it requires the use of detergents or lipids during crystallization. Membrane-spanning alpha helices are the most common structural motif in the membrane proteins. So, why is membrane proteins hard to crystallize for X-ray analysis? It's because crystallization of membrane protein is notoriously difficult, mainly because membrane proteins are extracted from their native phospholipid environment and transferred to detergents or membrane MIME-tech. In alpha helix, amino acid R groups protrude out from the helically coiled polypeptide backbone. Of the 20 amino acids, like alanine, glutamine, leucine, and methionine, are good alpha helix formers, but tyrosine, serine, glycine, and proline are not. The largely hydrophobic R groups of a membrane-spanning alpha helix contact the hydrophobic membrane core, while the more polar peptide backbone is buried. They consistently stabilize the helical conformation relative to glycine because it buries more polar area upon folding and because its backbone entropy is lower. They are also incompatible with alpha helix due to its rigid ring structure. Particular amino acids tend to occur at the different positions relative to the surface or interior of the bilayer in transmembrane segments of integral proteins. Residues with aliphatic side chains like leucine, isoleucine, alanine, valine predominate in the middle of the bilayer, which are insoluble or only slightly soluble in water. Aliphatic R groups are nonpolar and hydrophobic. Hydrophobicity increases with increasing number of carbon atoms in the hydrocarbon chain. Although these amino acids prefer to remain inside protein molecules, alanine and glycine are ambivalent, meaning that they can be inside or outside the protein molecule. Tyrosine and tryptophan are common near the membrane surface. It has been suggested that the polar character of the tryptophan amide group and the tyrosine hydroxyl, along with their hydrophobic ring structures, suits them for localization at the polar-apolar interface. These two are both amino acids, but they have different functions. Tryptophan can be used by the body in the process of making serotonin and melatonin, while tyrosine is used to produce noradrenaline and dopamine. Lysine and arginine are often at the lipid-water interface with the positively charged groups at the ends of their aliphatic side chains extending toward the polar membrane surface. Two of the polar amino acids like lysine and arginine contain amino-functional groups and are therefore positively charged. These two groups of amino acids are attracted to one another and can form electrostatic interactions. Cytochrome oxidase is an integral protein whose intramembrane domains are mainly transmembrane alpha helices. A 20 amino acids alpha helix just span a lipid bilayer. Hydropathy plots are used to search for amino acid stretches of hydrophobic amino acids in the primary sequence of a protein for which a crystal structure is not available. Putative hydrophobic transmembrane alpha helices have been identified this way in many membrane proteins. Hydropathy plots alone are not conclusive. Protein topology studies are used to test the transmembrane distribution of proteins domains predicted by hydropathy plots. If a hydropathy plots indicate 120 amino acids hydrophobic stretch, 1 putative transmembrane alpha helix, topology studies are expected to confirm location of N and C termini on opposite sides of membrane. If two membranes alpha helices are predicted, N and C termini should be on the same side. The segment between the alpha helices should be on the other side. Transmembrane topology is tested with impairment probes added on one side of a membrane. For example, protease enzymes. Degradation of protein segments indicates exposure to the aqueous phase on the side of the membrane to which a protease is added. Monoclonal antibodies raise to peptide equivalent to individual segments of the protein. Binding indicates surface of exposure of a protein segments on the side to which the AB is added. Such studies have shown that all copies of a given type of integral protein have the same orientation relative to the membrane. Flip-flop of integral proteins does not occur. Transmembrane topology is tested with impairment probes added on one side of a membrane. For example, protease enzymes. Degradation of protein segments indicates exposure to the aqueous phase on the side of the membrane to which a protease is added. Monoclonal antibodies raise to peptide equivalent to individual segments of the protein. Binding indicates surface exposure of a protein segments on the side to which the AB is added. Such studies have shown that all copies of a given type of integral protein have the same orientation relative to the membrane. Flip-flop of integral proteins does not occur. A helical wheel looks down the axis of an alpha helix, projecting side chains onto a plane. An alpha helix lining a water-filled channel might have a polar amino acid R groups facing the lumen and non-polar R groups facing lipids or other hydrophobic alpha helices. Such mixed polarity would prevent detection by a hydropathy plot. Foreign B-barrel white transmembrane alpha helices are the most common structural motif for integral proteins. A family of bacterial outer envelope channel proteins are called porins, have instead B-barrel structures. A B-barrel is a B-sheet rolled up to form cylindrical pore. In a B-sheet, amino acid R groups alternately point above and below the sheet. Much of porin primary structures consist of alternating polar and non-polar amino acids. Polar residues face the aqueous lumen. Non-polar residues are in contact with membrane lipids. Structures of lipids. Lipids or fats are composed mainly of carbon, hydrogen, and oxygen. Some lipid contains other elements such as nitrogen and phosphorus, and most lipids do not dissolve in water because they are non-polar, meaning that electrons are shared equally in the molecule, so they have no partially charged regions in the molecules. The main types of lipids include triglyceride, phospholipids, steroids, and prostaglandins. Triglycerides are the most plentiful lipid in the body, and they are composed of two building blocks, a glycerol unit and three fatty acid chains. A glycerol molecule has 3 carbon atoms with 2 bonded to 2 hydrogen atoms and 1 bonded to 1 hydrogen atom. They are also bonded to a hydroxyl group, which is an oxygen bonded to a hydrogen, and the fourth bond being to another carbon atom. Fatty acids are made up of long chains of carbon atoms and hydrogen atoms with an end carbon atom bonded to a hydroxyl oxygen group and a double bond to an oxygen atom. There are 2 types of fatty acids, saturated fatty acids and unsaturated fatty acids, which include monounsaturated fatty acids, polyunsaturated fatty acids, and trans fatty acids. Triglycerides are found in many food items such as vegetable oil, coconut oil, beef, fast foods, avocados, and many different seeds. Triacylglycerols or TAGs. These are molecules with 3 ester groups, and examples include peanut oil, corn oil, butter, and lard. In general, fats like TAGs are used as energy storage. Glycogen only provide energy for about a day. Fats, energy for a month. Terpene is another type of lipid. A terpene consisting of 2 isoprene units. Farnesene found in apples has 3. The units can be linked to form rings such as limonin, which is found in lemon. These can get much larger, such as beta-carotene, comprised of 8 isoprene units, which is found in various plants and fruits. Terpenoids are similar to terpenes, but they contain oxygen atoms, such as with menthol, found in peppermint. These are just a few types of lipids found in our body. The fatty acid end is a nonpolar, so it is called hydrophobic, which means water-fearing. This property, having a hydrophilic and hydrophobic region, allowed this molecule to join or bridge a water environment and a lipid environment. So in water, these molecules will form bilayers, with the fatty acid tails facing each other and the heads facing outwards, as you can see here on the screen. Phospholipids are the main component of cell membrane. Phospholipids are similar in structures to triglycerides in that they have 4 subunits. They are composed of glycerol unit, 2 fatty acid chains, a phosphate group, and a nitrogen-containing group. The phosphate of the nitrogen extend in the opposite direction of the fatty acids. This end or head of the molecule is actually polar, so it is a water-soluble. Hydrophilic, meaning water-loving, is the term often used to describe the head portion of the phospholipid molecule. Prostaglandins are lipids that are derived from unsaturated fatty acids. These are composed of 20 carbon, 5 carbon ring structures. There are many different types of prostaglandins in the body that take part in different important functions, such as enhancing the immune system and inflammatory response. Steroids, which are often referred to as sterols, have 4 hydrocarbon ring or steroid nucleus at the foundation of their molecules. Steroids differ based on the side chains extending out from their rings. Cholesterol, whose molecular model is shown here, is an important steroid as it has hydrophilic, water-loving polar region here at the hydroxyl oxygen end of the molecule, and hydrophobic, water-fearing nonpolar region at its hydrocarbon chain. Cholesterol is a key part of cell membranes as its polar region can interact with the polar region of the phospholipids, and its nonpolar region is embedded in the membrane alongside the nonpolar fatty acid of the phospholipids. Cholesterol is also important building blocks in bile acids, which are the key in breaking down fat molecules in the body, and many hormones, which are signaling molecules the body releases. Cortisol, estrogen, and testosterone are also other important steroid compounds. You can see that each of these molecules differ slightly based on their side chain formation, but they have all 4 hydrocarbon rings as the foundation of their molecules.
Good morning, my dear teacher and classmates. By the way, my name is Lester Mike Vergado, and for today I'm going to tell you about steroids. So when people hear the word steroids, they typically think of anabolic steroids, which are synthetic derivatives of testosterone. But in actuality, steroids refer to a class of molecules that includes not just hormones, but also certain vitamins and cholesterol. So let's learn a bit about the structure and function of steroids. Steroids are all derivatives of this ring system, which is comprised of 4 rings. The A, B, and C rings are 6-membered rings, while the D ring is a 5-membered ring. There are also always metal groups on these 2 carbons, and for ease of communication, we'll always number this ring system the following way. So this is carbon 1, and numbers 1 to 10 will wrap around rings A and B this way. The carbon 11 jumps over to ring C, and we wrap around ring C and D. Lastly, numbering these 2 metal groups like this. All the steroids we examine will be based on this structure. Now we know that 6-membered rings like to be in chair conformations, so let's see what that would actually look like. First, we should note that there are 2 common conformations for the A ring, either with a trans ring junction between A and B like this, or a cis junction putting First, you should note that there are two common conformations for the A ring, either with a trans ring junction between A and B like this, or a cis junction putting a ring A like this. Also notice that the metals are in the actual position, pointing upwards, and we will commonly refer to the substituents on the same side as those metals as beta substituents, whereas ones on the other sides, trans to the metals, will be alpha substituents. The orientation of a ring will depend on the hydrogen on carbon 5. If that hydrogen is alpha, or on the dashed bond, you get the trans AB junction. If the hydrogen is beta, or on the wedge bond, you get this cis AB junction. Now, where else do steroids vary in structure? A common location for our substituents is carbon 17. Different alkyl groups at this location will generate different steroids, one of which is cholesterol. Cholesterol has 8 stereocenters, which means 256 different stereoisomers of this structure are possible, but only this specific one is cholesterol. This is an intermediate in the biosynthesis of all the steroids we need in our body. And although we often consume cholesterol, we don't need to, because we also make it ourselves. So we will typically have more cholesterol than we need. And too much cholesterol in the diet leads to clogged arteries and heart problems. So let's look at another type of steroids. The sex hormones. This can be divided into 3 classes: the female hormones or estrogens, the male hormones or androgens, and pregnancy hormones or progestin. And now let's look at the female hormones first. These are estrogen and estradiol, which differ only in the level of oxidation of carbon 17. Estradiol is secreted by the ovaries and kicks off the changes that occur during puberty, and also causes development of the mammary glands during pregnancy, whereas estrogen is just a metabolized form of estradiol that is excreted. The male sex hormones and androsterone and testosterone are incredibly similar to the female hormones, with the only differences occurring in the A ring. Similarly to the female hormones, testosterone does all the work promoting the growth of body hair and facial hair, lowering of the voice and other traits associated with puberty, whereas androsterone is just the metabolized version that is excreted into the urine. And progesterone is the most important pregnancy hormone, which prepares the uterine lining for implantation of fertilized egg and suppresses further ovulation during pregnancy. Other hormones like cortisol and cortisol regulate many other biological activities like metabolism and controlling inflammation. And that wraps our survey of lipids. Thank you.
Good morning, ma'am and classmates. My name is Tata Baikanda, and I'm going to tell you about the nature of enzymes. When an enzyme's environment changes either to hot or a big change in pH salt, concentration or solvent type, the process called denaturing, the word enzyme amines in yeast where they were first discovered hides on one of the enzymes in yeast is able to catalyze the conversion of sugar into alcohol and carbon dioxide. Carbon dioxide is reaction helps bread due to rice and the ethanol called be the biofuel for Brazilian cars or called be used to make alcoholic drinks. Perhaps the communist example of denaturing the protein is when you cook an egg. The while of an egg is made of soluble protein molecules called albumin, we use to preserve food for a long time. The main methods are salting, eating, preserving in acid, like pickling, preserving in alcohol, like wine, freezing, or also preserve food by drying methods. Do not denature enzyme but work by keeping out water in which mold and bacteria thrive. So will the primary structure of proteins is with amino acid. There are 22 choice from join with the peptide bond. The coil's structure of proteins is with amino acid. There are 22 choice from join with the peptide bond. The coils into a sort of chain held together by the attraction between the amino and carboxylic acid groups, forming their secondary structure with different side groups of the amino acid. The protein, the coil up in a very specific third shape is tertiary, causing the enzyme the unravel with radius. This happens at around 45 degrees Celsius. But denature their enzymes similarly and increase in concentration of ions such as hydrogen, hydrazide, or salt, or changing the solvent from water polar to alcohol will disrupt this default forces, causing the enzyme to methods of food preservation work by denaturing the enzyme in the mold and the bacteria. Enzymes work constantly until they are dissolved or become denatured. When enzymes denature, they are no longer active and cannot function. Extreme temperatures and the wrong levels of pH, a measure of substances activity, acidity, or alkalinity, can use enzymes to become denatured.